The current video is regarding the reasons for bioequivalence failure. This video will discuss regarding understanding the reasons behind the bioequivalence study failure. Also, this video will discuss for the causes and solutions. Also, in this video we are going to learn regarding key challenges in bioequivalence studies for the generic drugs. Welcome to the video, and I request you to be with this video till the end, and I assure that you will get good knowledge about the bioequivalence studies failures in this video. Generic drugs are required to be bioequivalent to the reference formulations, and bioequivalence studies provides the information regarding the similarity of the therapeutic effect between the reference formulation and the generic formulation. Bioequivalence studies demonstrate that the generic formulation or generic product is having the same pharmacokinetic properties as per the reference drug or RLD or the comparator product or the marketed product. It is also known as reference formulation. And bioequivalence demonstration is very much required and is essential for the patient safety and also for the regulatory approval. Without demonstration of the bioequivalence, the generic product cannot be approved. Why bioequivalence is important? Because it ensures safety, efficacy, and therapeutic interchangeability of the generics. After demonstration of the bioequivalence, the reference formulation can be changed with the generic formulations, and this will have impact onto the costing of the medicines. It also avoids the research costs, also avoids the delays in the drug availability to the public, and it also avoids the erosion of public confidence. If the generic is not bioequivalent, then there is no approval. Reasons for bioequivalence failures. So, bioequivalence means similarity in the rate and extent of the drug absorption and pharmacokinetics. Now, why this fails or why there is a failure in the bioequivalence study? The reasons are many and depend on case by case basis, but some of them and important reasons I have enlisted here. The reason is high variability in the drug absorption and the metabolism. Inadequate bioequivalence study design. There may be some differences in the formulations. There are challenges in the regulatory requirements. Patent may be there, because of which the generic formulations are made in a non-infringing way, and because of that the bioequivalence failures may happen. Then patient population issues might be there, analytical methodology problems may be there, and some manufacturing problems and issues might occur. Variability in the drug absorption and metabolism. As we know, the ADME of the drug is affected because of the genetic differences. CYP450 enzymes variations are there between the individuals, which affect the drug metabolism, and poor and ultra rapid metabolizers cause wide fluctuation in the drug exposure. If there is first pass metabolism, it may vary. Enzyme variations may impact the drug metabolism, and that's why may impact the pharmacokinetics. Second is narrow therapeutic index drugs or NTI drugs. So NTI drugs are required to be bioequivalent with a stringent criteria is there. Small change in the drug exposure can have severe clinical effects. Example is warfarin, other is digoxin. phenytoin. So all these are the narrow therapeutic index drugs, and because of the small changes in drug exposure, the bioequivalence may fail. Now coming to the physiological factors like age, gender, BMI, comorbidities in the patient population can influence drug pharmacokinetics. Inadequate study design. Sometimes the bioequivalence study design is itself having a fault or problem, and that's why there might be a chance of bioequivalence failure. If small sample size is there, like subjects, insufficient power to detect real differences between the generic and reference. So because of the small sample size, there may be failure. Improper dosing or improper dosing timing may lead to incorrect dosing regimen, and inadequate washout periods can result into the failure results. Then failure to account for variability. So if variability is there and if it is not calculated or checked, then there are high chances of the bioequivalence failure. High intrasubject variability in absorption, metabolism needs proper study design. Formulation differences might be there, differences in the physicochemical variations, differences in the composition, excipients, their grades and levels, changes in the particle size of the API, and method of manufacturing may lead to change in the absorption. All these formulation differences may lead to change in the dissolution, and it will lead to changes or variation in the absorption. Polymorphic drugs, different crystalline form of the same drug, may result in distinct pharmacokinetics. As per my understanding and the experience, if you select different polymorph than the innovator drug polymorph, then there are chances of bioequivalence failure because the solubility is different, stability is different, and absorption is different for different crystalline polymorphs. Regulatory challenges. There are some regulatory challenges like different guidelines as per the regions, FDA versus EMA. So example is FDA versus EMA guidelines. So these differences may lead to inconsistent results. Then tight bioequivalence limits. So drugs with narrow therapeutic index have strict B criteria that uh, may make equivalence harder to demonstrate or prove. For NTI drugs, the bioequivalence criteria is 90 to 111%. The geometric mean for Cmax and AUC T/R ratio should be between 90 to 111% for NTI drugs. So it makes it difficult to prove the bioequivalence. Patient population and study inclusion. This is another challenge, another problem, or another reason for the failures. Non-representative study population. Excluding diverse patient groups can skew the results. In exclusion of critical subgroups, excluding patients with renal liver impairment or other comorbidities can affect the validity because all these type of factors affect the pharmacokinetics. Analytical and methodological problems. Sometimes there may be problem associated with the analytical errors and the manufacturing issues. Inaccurate or inconsistent methods for measuring drug concentrations can lead to false conclusions. Sensitivity of testing methods. Some drugs require more sensitive tests to detect small but clinically relevant differences. Manufacturing issues may be there. Quality control problems, batch to batch variability in manufacturing can impact the drug consistency and bioequivalence. You might have observed regarding the dissolution studies. So if unit to unit variations are there, these variations may lead to problems in the bioequivalence. Production inconsistencies. Issues in manufacturing processes can lead to differences in drug formulation, affecting the bioavailability. Analytical and manufacturing errors or problems may have greater, much and more impact onto the bioequivalence. Now coming to the solutions. So what are the solutions? Solutions are dependent on case by case basis, and all the solutions are not generalized. These are dependent on to the formulation design, molecule type and criticality of the bioequivalence, and a risk associated with the bioequivalence studies. Some of the generalized solutions I have given here, like enhanced study designs, pharmacogenetic screening, and pre-study simulation. Use of larger sample size may sometimes solve the problem. Replicate study designs, maybe partial replicate or full replicate, may solve the problem. And advanced statistical models, Bayesian methods, can give you the good answer to the bioequivalence failure. Incorporating genetic data to predict drug metabolism and reduce variability. Nowadays, simulations are being done. In vivo simulation can be done, and based on that, some problems can be solved or understood. Pre-study simulation, modeling and simulation to optimize protocols before conducting the study. Also, before conducting a full study, sometimes pilot study will give you the indication for would be success or failure. Summary of the bioequivalence failures we will see now. Bioequivalence failures may arise from the different problems or factors, may be related to the formulation, may be related to the design, subjects, and many more. Sometimes combination problems may be there, like combination of physiological parameters, design, formulation, and the regulatory challenges. So, future direction will be embrace personalized medicine and advanced statistical model to reduce the variability. And importance of addressing these issues is ensuring successful bioequivalence studies, which is crucial for safe and effective use of generic drugs. The solutions may differ from case by case basis, product to product. Sometimes increasing the sample size may solve the problem. Sometimes changing the study design may solve the problem. Sometimes you need to reformulate the product to meet the bioequivalence criteria. There are different problems, there are different solutions, and these all are dependent on to the formulation type and the criticality in the bioequivalence. Complex design formulations and solubility enhanced formulations always show problem in the bioequivalence. So always try to match the quality of the generic formulation, composition of the generic formulation to the reference formulation, and then you can have some confidence to go for the bioequivalence studies, and you will have less chances of the failures. I hope this video is providing you the good idea for the bioequivalence failure reasons, and I hope that this video will be helpful to you to have good understanding for the bioequivalence studies. If you have any queries, please write in the comments and I will try to solve your questions. Thank you for watching the video.